Megazyme provides an extensive range of assay kits for use in various assay formats, including autoanalyzer, microplate, and manual spectrophotometer. This lactose test kit, K-LOLAC, is the only enzymatic method commercially available for the rapid, quantitative, sequential, and accurate measurement of free glucose and lactose in conventional, low-lactose, and lactose-free dairy products. This video tutorial will demonstrate the use of the kit with the low-lactose milk sample and the kit standard using the manual spectrophotometer format. The kit contains sufficient reagents for 65 manual assays and is supplied with a detailed data booklet. This assay specifically measures D-glucose, and through the use of a selective beta-galactosidase, MZ104 beta-galactosidase, it is highly selective for lactose. The principle of the enzymatic reaction involved in the measurement of lactose is shown in this figure. Pre-treatment of low-lactose and lactose-free samples is required to efficiently remove free D-glucose from the sample. This is carried out by converting the free D-glucose to D-gluconic acid by the enzymes glucose oxidase and catalase in the presence of oxygen. Prior to lactose hydrolysis, any remaining free D-glucose is phosphorylated by hexokinase in the presence of ATP to glucose 6-phosphate plus ADP. Glucose 6-phosphate is oxidized by the enzyme glucose 6-phosphate dehydrogenase in the presence of NADP+ to gluconate 6-phosphate with the formation of NADPH. Gluconate 6-phosphate is immediately converted to D-ribulose 5-phosphate, carbon dioxide, and a further molecule of NADPH by the enzyme 6-phosphogluconate dehydrogenase. This step is added as it increases the sensitivity of the assay twofold. Subsequently, lactose is hydrolyzed to D-galactose and D-glucose by beta-galactosidase MZ104. Glucose released from lactose enters the series of reactions catalyzed by hexokinase, glucose 6-phosphate dehydrogenase, and 6-phosphogluconate dehydrogenase. The amount of NADPH formed in these reactions is stoichiometric to twice the amount of lactose, as 2 molecules of NADPH are produced for each D-glucose molecule originating from the lactose in the sample. It is the NADPH formation which is measured by the increase in absorbance at 340 nanometers. Prior to sample analysis, prepare the required additional reagents as described in the kit data booklet. Subsequently, once all of the additional reagents have been made, the kit components should be prepared, and once prepared, they are ready for use in the manual assay procedure. Use the contents of bottles 1, 3, 5, 6, and 7 as supplied. The contents of bottle 2 are dissolved in 14 milliliters of distilled water. The bottle is capped and the contents are mixed thoroughly. Divide into appropriately sized aliquots and store in polypropylene tubes below -10 degrees Celsius. The contents of bottle 4 are dissolved in 4 milliliters of distilled water. The bottle is capped and the contents are mixed thoroughly to ensure complete dissolution. Divide into appropriately sized aliquots and store in polypropylene tubes below -10 degrees Celsius. Follow the sample preparation method as described in the data booklet. In this demonstration, we are using a low-lactose milk sample in duplicate, sample A. Clarify the milk sample as follows. Pipette 0.9 milliliters of distilled water to microfuge tubes in duplicate. Add 0.5 milliliters of milk sample to both tubes. Add 0.05 milliliters of concentrated Catalase 2 solution. Add 0.05 milliliters of concentrated Catalase 1 solution. Cap the tubes, mix by inversion and vortex, and then centrifuge at 13,000 RPM for 10 minutes. Prepare the sample for free D-glucose removal as follows. Add 0.4 ml of distilled water to 13 ml polypropylene tubes in duplicate. Add 1 ml of clear supernatant from each tube from the clarification step. Add 0.1 ml of bottle 1, buffer A. Add 0.2 ml of prepared bottle 2, glucose oxidase catalase mixture. Add 0.1 ml of hydrogen peroxide, 30% weight per weight. Immediately cap the tubes. Mix the contents on a vortex mixer and incubate at room temperature for 15 minutes to allow oxidation of glucose to gluconic acid. After 15 minutes, slowly loosen the cap to release pressure and retighten. Incubate tubes in a 100°C water bath for 5 minutes to inactivate the glucose oxidase and catalase. After 5 minutes, remove the tubes from the water bath. Transfer the solutions to labelled 2 ml microfuge tubes and centrifuge at 13,000 RPM for 10 minutes. Carefully transfer the clear supernatant from the centrifuge samples into clean, labelled microfuge tubes for use as the sample solution in the enzymatic determination reaction. In this demonstration, we will use the prepared, pre-treated, low-lactose milk sample in duplicate and the lactose standard solution, bottle 7, along with a reagent blank. Accurately pipette 1 ml of distilled water into the blank tube, 0.9 ml into the sample A tubes, and 0.9 ml into the standard tube. Accurately pipette 0.1 ml of the pre-treated sample solution into the sample A tubes. Accurately pipette 0.1 ml of the lactose standard solution, bottle 7, into the standard tube. Accurately pipette 0.1 ml of buffer B, bottle 3, into all tubes. Accurately pipette 0.05 ml of NADP plus and ATP, bottle 4, into all tubes. When all of the components have been added, mix the tube contents thoroughly and incubate the tubes at 25°C for approximately 3 minutes, either in the onboard incubator on the Megazyme Wave or in a separate incubation block. After 3 minutes, record the first absorbance reading A1 at 340 nanometers for all of the assay tubes. In this demonstration, we are using the Megazyme Wave spectrophotometer, set to read at 340 nanometers. Alternatively, a recording spectrophotometer with 1 cm path length cuvettes can be used. Swirl to mix the contents of bottle 5 hexokinase plus glucose 6-phosphate dehydrogenase plus 6-phosphogluconate dehydrogenase suspension prior to dispensing. Pipette 0.02 ml of bottle 5 into all assay tubes. Mix the tube contents thoroughly and incubate the tubes at 25°C for approximately 10 minutes. After 10 minutes, record the second absorbance reading, A2, at 340 nanometers for all of the assay tubes. Swirl to mix the contents of bottle 6 beta-galactosidase prior to dispensing. Pipette 0.02 milliliters of bottle 6 into all assay tubes. Mix the tube contents thoroughly and incubate the tubes at 25 degrees Celsius for approximately 15 minutes. After 15 minutes, record the third absorbance reading, A3, at 340 nanometers for all of the assay tubes. This absorbance reading completes the lactose assay procedure. The relevant absorbance readings A1, A2 and A3 of the sample and the blank reactions are used to calculate the lactose concentration in the original samples. When performing this test using the pre-installed protocol on the MegaQuant-Wave spectrophotometer, the results will be automatically calculated and printed via the onboard printer, or the data can be exported to a computer using the SF Capture software. Please see our MegaQuant-Wave video for further details. If the results output are raw absorbance values for both blank and samples, the calculations of lactose content can be performed manually as described in the calculation section of the kit booklet. Megazyme has also developed specific Excel-based MegaCalc applications for each Megazyme kit to allow quick and easy results analysis. Results can be analysed using the MegaCalc application specific to this lactose assay kit, which is available to download free of charge from the Megazyme website. The MegaCalc spreadsheet provides full instructions for use. Open the MegaCalc worksheet and input the following: sample details, absorbance readings for the blanks. For each sample, input the sample identifier. For each sample, input the absorbance values recorded. If a reaction creep has been identified, enter this data on the Creep Calculation MegaCalc tab. Once this data is entered, it will automatically transfer into the A3 Creep Corrected column on the main MegaCalc tab and be taken into consideration in the final calculations. Alter the sample volume if a volume other than the default 0.1 milliliters is used. Alter the dilution if a dilution other than 5.4 is used. This is the dilution for the standard preparation for liquid milk. When all of the data has been entered, the concentration of lactose in the sample is automatically calculated and given as grams per litre in the original sample. For solid samples, input the concentration of the original sample extract in grams per litre. The concentration of lactose in the solid samples is then automatically calculated and provided as grams per 100 gram in the original sample. This novel enzymatic method for the measurement of lactose and lactose-free products is discussed in detail in a paper by Mangan et al. This method has also recently been approved for first action status as an AOAC single laboratory validated method.